Welcome to Tomorrow Unveiled.
It is good to be here.
It is. And I have to say, I am still trying to process the stack of papers you sent over last night.
This is a big stack.
It's a very big stack. Usually when we sit down to do one of these, we're looking at, incremental progress. A mouse lived maybe 5% longer, or we found a new vitamin that might help with skin.
Right. Nice things.
Nice stuff, exactly, but not earth-shattering.
Incrementalism is how science usually works. It's a slow, steady grind.
Exactly.
Yeah.
But the week of January 21st to January 28th, 2026, this doesn't feel like a grind. It feels like the dam broke.
That is honestly the perfect metaphor. I genuinely believe that when historians of science look back at this decade, they're going to circle this specific week in red ink. We are seeing a definitive inflection point.
An inflection point. That's a heavy term.
Yeah.
You're putting this week up there with, the discovery of penicillin, the splitting of the atom.
I don't use the comparison lightly. I mean, look, for the last 20 years or so, longevity science has been kind of stuck in what I'd call the worm and yeast phase.
Right.
We've gotten incredibly good at making nematodes live longer. We've been great at extending the lifespan of fruit flies. But the translation to humans, that has always been the valley of death for biotech.
I've heard that phrase.
It works in the lab, it fails in the clinic.
And we've all seen that headline 1,000 times. Cancer cured in mice.
Exactly. But this week, the bridge across that valley just got built. I mean, we aren't just talking about speculative papers anymore. We are talking about FDA clearances. We're talking about human safety data.
Which is huge.
And maybe, most importantly, we are seeing a shift in the philosophy of the science itself.
How so? What do you mean by that?
Well, we aren't just talking about lifespan anymore. The goal isn't just to tack on extra years at the end of life where you might be frail, suffering, or, bedridden.
The nightmare scenario.
That is the absolute nightmare scenario. It's the Tithonus error from the Greek myth. You get eternal life, but you also get eternal aging. The data that dropped this week is squarely focused on functional life extension.
Which is the only kind that actually matters. I don't want to be 120 if I can't tie my own shoes or remember my grandkids' names.
Of course not.
I want to be 100 and feeling like I'm 40. I want the hardware to work.
Precisely. We are moving from the era of, biohackers mixing questionable compounds in their garages to rigorous, major medical institution clinical reality. We're talking about engineering our biological limits.
Okay, so we have a massive amount of ground to cover. Our research team has been working overtime, pulling everything from academic journals to FDA filings. We need to structure this so our listeners don't get vertigo. How are we breaking this down?
So we've organized this into what I call the 3 pillars of this new era. It's the clearest way to see it.
The 3 pillars. I like it.
First, we have resetting the clock. This is the headline event, the first human trial for epigenetic reprogramming.
Turning old cells young again. The holy grail.
Correct. The big one. Second, we have taking out the trash. We have major updates on senolytics, clearing out those toxic zombie cells. And finally, some real safety data to back it all up.
Which is crucial, because up until now, that safety data has been, let's call it murky.
Murky is generous, but we have clarity now. And third, pillar number 3, we have the maintenance crew. This covers gene editing, innovative new ways to mimic exercise, and blocking the inflammation loops that degrade us over time.
It sounds like science fiction, but the documents in front of me say otherwise. Start with the big one, the software reset.
Let's do it.
Okay, let's unpack this. The term in the report is ER100. Our dedicated research team highlighted this as the biggest news of the week, maybe the year. The FDA has cleared the first ever human clinical trial for partial epigenetic reprogramming.
This is monumental. I mean, really, this is the moment we go from can we do it in a dish to we're doing it in a person.
For those listening who might not be, PhD candidates in biology, we need to really break down what epigenetic reprogramming actually means. I love the analogy you use, the computer hardware versus software, but let's go a little deeper.
It's absolutely the best way to visualize it. Think of your DNA, your genome, as the hardware. It's the hard drive. It contains all the instructions for building you. That hardware is extremely stable. Barring some, radiation damage or random mutations, it doesn't change much over your lifetime.
So my DNA is basically the same when I'm 5 as when I'm 80.
Essentially, yes. But then you have the epigenome, and this is the software, the operating system that runs on top of the DNA. It's what tells the cell which genes to turn on and which genes to turn off.
Because every single cell has the same DNA, right? My skin cell has the code for my liver, and my liver cell has the code for my eyeball.
Exactly. But the software, specifically things like methylation tags, these little chemical markers, tells the skin cell, hey, you run the skin code and just ignore the liver code. It's what differentiates the cell and gives it its job.
Okay, so what happens when we age? Does the software crash?
It gets corrupted. That's the perfect word. Imagine a CD getting scratched or a computer operating system that hasn't been rebooted in, I don't know, 80 years.
It's going to be slow and buggy.
Very buggy. You install updates, you uninstall programs, you browse the web. Eventually, the registry gets messy. In biological terms, this epigenetic noise means genes that should be silent start waking up, and genes that should be active start shutting down.
So my skin cell starts trying to be a little bit of a liver cell, or it just gets confused.
Or it just forgets how to be a competent skin cell. It loses its identity. And this loss of information is now understood to be a primary driver of aging.
And ER100 is the reboot button, the Control-Alt-Delete for the cell.
That is the promise.
Right.
The science goes back to the Nobel Prize-winning discovery of the Yamanaka factors. 4 specific transcription factors, these are proteins, that can take an adult cell and revert it all the way back to a stem cell.
Okay, but let's double-click on this for a second. Because I recall reading about the early Yamanaka experiments, and they weren't exactly trying to save eyesight or make people look younger. They were trying to turn skin cells into stem cells in a petri dish.
That's right. It was a basic science tool.
So if you just blast a living human with these factors, doesn't the cell just forget what it is entirely?
That is the inherent risk. And it's a huge one. It's called de-differentiation. If you run the software update for too long, or way too aggressively, a skin cell completely forgets it's a skin cell and reverts to a primordial pluripotent state.
And why is that bad? Stem cells sound good on the surface.
In a petri dish, it's a triumph. In a human body, it's a complete disaster. It's a tumor waiting to happen. You'd be a blob of undifferentiated tissue, or you'd develop teratomas.
Teratomas, right.
Tumors made of teeth and hair and muscle all mixed together. It's horrifying.
Right, you definitely don't want your optic nerve suddenly deciding it wants to be a tooth. That's body horror territory.
Exactly. So the real breakthrough with ER100 isn't just the cocktail of factors. It's the timing and the composition of that cocktail. The original Yamanaka cocktail was 4 factors: Octave4, SOX2, KLF4, and C-Mike.
And the mic part is the one our notes flagged in big, bold, red letters.
Red letters for a very good reason. C-Mike is a potent oncogene. It basically hits the gas pedal on cell division. It's great for making a lot of stem cells quickly in a lab, but it's terrible for safety in a patient.
So what do?
The team behind ER100 made a calculated engineering decision. They dropped C-Mike entirely, just took it out of the equation.
Does that weaken the effect? I mean, I assume C-Mike was there for a reason in the first place.
It slows the process down for sure, but in this context, slowness is a feature, not a bug. By removing C-Mike, they created a much gentler slope of reprogramming. It allows the cell to erase the epigenetic markers of aging, those methylation tags that say I am old, without erasing the methylation tags that say I am a retinal ganglion cell.
So it's a partial reset. The skin cell remembers it's a skin cell, it just thinks it's a young skin cell.
Exactly. It's restoring the function without losing the identity. That's a key.
And they're starting with the eye, specifically optic neuropathies like glaucoma. Why the eye? Why not say the skin or the liver or the heart? Those seem like bigger targets.
Several very strategic reasons. From a scientific perspective, the eye is what we call an immune-privileged site. It's somewhat isolated from the rest of the body's immune system, which makes it a much safer place for testing new genetic therapies. If something goes wrong, it's generally contained.
That makes sense. A firewall?
Sort of, yes, but even more importantly, it offers measurable functional endpoints.
You can just ask the patient, can you see the chart better?
Immediately. You can measure it. You don't have to wait 10 years to see if they die or not. In the animal studies leading up to this, and our research team looked at the primate data specifically, they didn't just look at cells under a microscope. They restored vision in primates that had lost it due to glaucoma-like damage. So if this trial shows that ER100 can restore visual function in humans by making the retinal ganglion cells younger, it validates the entire concept.
That's the aha moment for me. If it works in the eye, it proves that tissue age is actually reversible. Not just slowable, but reversible.
It moves rejuvenation from a theoretical, almost philosophical concept to a verifiable clinical outcome. We aren't just slowing down the decline of vision, we are potentially winding the clock back to restore function that was lost. It's a paradigm shift from treating symptoms like, say, lowering eye pressure and glaucoma to fixing the root cause. the age of the cell itself.
But I have to play the skeptic here for a second. The eye is a small, contained environment. Just because we can reboot the eye, does that really mean we can reboot the whole body? Or are we just going to have 90-year-old bodies walking around with 20-year-old eyes?
That is the billion-dollar question. And you're right to ask it. Systemic delivery, getting this reboot to every cell in the body safely without triggering an immune response or cancer, that is a much, much harder engineering challenge. But proving the biological principle in the eye is step one. If the biology holds, the engineering will follow.
It's the Wright brothers moment. Even if the first flight is only 12 seconds and 120 feet, it proves flight is possible.
Precisely. This is that moment for rejuvenation.
All right, let's move to our second pillar. If reprogramming is about resetting the software, this next part is all about taking out the trash.
Senolytics.
Zombie cells. I feel like this term gets thrown around a lot in pop science, and it's almost lost its meaning. But the updates from this week suggest we are finally getting past the hype. First, let's redefine the enemy. What is a senescent cell, really?
Okay, so a senescent cell is a cell that has reached the end of its division cycle or has been damaged by stress. Normally, a cell in that state should do the polite thing and commit suicide, a process called apoptosis, and then get recycled by the immune system.
Taking one for the team.
Right. It's a clean exit. But a senescent cell refuses to die. It enters this state of suspended animation. It's not dead, but it's not functioning properly, hence zombie.
But if it just sat there doing nothing, that wouldn't be so bad, would it? It would just be a microscopic squatter taking up space.
If only. These aren't passive zombies. They are metabolically active and, frankly, incredibly malicious. They secrete that toxic cocktail of inflammatory signals we mentioned, the SASP, the senescence-associated secretory phenotype.
SSP. Sounds nasty.
Think of it like a rotten apple in a fruit bowl. That one rotten apple releases ethylene gas that spoils all the healthy fruit touching it.
Yeah.
Senescent cells release these inflammatory cytokines that damage the healthy cells around them, and, this is the really key part, can actually turn those healthy neighbors into zombie cells too.
It's contagious aging.
Locally, yes, it spreads the damage, so we desperately want to get rid of them. But the problem has always been finding them. They are incredibly heterogeneous. They look different depending on where they are in the body. A zombie fat cell looks different than a zombie brain cell. There's no single zombie name tag.
So how do we target them? Our notes mention a breakthrough using something called DNA aptamers.
This is a very cool piece of technology. Aptamers are short strands of synthetic DNA or RNA, but they aren't used for coding genes. They are designed to fold into complex, very specific three-dimensional shapes.
Like molecular origami.
Exactly like molecular origami. And because shape determines function in biology, you can design an aptamer that fits perfectly, like a key in a lock, into a specific protein on the surface of a senescent cell.
So it's a homing beacon.
Or a highlighter pen. This new technique our team flagged allows researchers to tag these zombie cells in living tissue with incredibly high precision. It means we can finally visualize the burden of senescence in a living organism. You can't treat what you can't measure. Now we see exactly where the damage is accumulating.
And speaking of treatment, let's talk about the drugs. We have some actual human safety data this week from a big collaborative study. They used the classic combo, dasatinib plus quercetin.
T and Q, the granddaddy of senolytics.
But let's pause on the dasatinib part of that equation.
Yeah.
Because I looked this up, and dasatinib isn't a vitamin. It's a tyrosine kinase inhibitor used for leukemia. It has a black box warning from the FDA. Are we really suggesting that healthy people or even just older adults with, say, mild cognitive impairment should be taking a chemotherapy agent? That sounds scary.
It's a fair hesitation, and it's absolutely the reason the field has moved so cautiously. If this were a daily pill, the answer would be a flat no. The side effect profile, fluid retention, anemia, immune suppression is real, and it is dangerous.
Right, I don't want to trade my memory loss for heart failure.
And that is where the biology of senescence dictates the protocol. This is so important. Remember, we aren't trying to change a metabolic pathway that runs 24/7, like blood pressure. We are trying to kill a specific population of cells. Once a zombie cell is dead, it stays dead.
It doesn't respawn the next day.
Not immediately. It takes weeks or even months for new senescent cells to accumulate from metabolic stress. And this allowed the research teams to design what they call a hit-and-run protocol.
Hit and run, I like that.
The theory is that you don't need the drug in your system constantly. You go in, you clear the accumulated zombie cells, and then you get out. You let the body recover. In the study, they administered the drugs for just 2 days every 2 weeks. a quick pulse.
You clearing the trash and then letting the sanitation crew go home for a couple weeks.
Precisely. And in the safety data, this intermittency was absolutely key. The adverse events in the treatment group were statistically indistinguishable from the placebo group. That tells us the toxicity requires chronic, daily exposure, but the benefit, the senolysis, the cell killing, happens acutely in that short window.
That's a massive relief. It validates that we don't need to bathe our cells in these toxins to get the benefit.
It makes the therapy viable for the first time in a real-world setting, but there's an evolution happening here too. We are moving from carpet bombing with broad-spectrum drugs like dasatinib to precision strikes.
And this brings us to the antibody-drug conjugates, or EDCs. We saw news about a collaboration involving something called STX1.
Yes, this is fascinating. They are borrowing technology directly from modern cancer treatments. Instead of a drug that just floats around the whole body and hits everything, they attached a toxin to an antibody.
The antibody is the delivery truck.
And it's a delivery truck with a very specific GPS-guided address. It targets a protein called CD26 or DPC4, which is found abundantly on the surface of senescent cells, but not on most healthy cells.
So it's a laser-guided missile.
Exactly. It delivers the toxic payload only to the bad cells, sparing the healthy ones.
Which should drastically reduce side effects even further.
And increase potency. You can use a much stronger toxin because you know it won't touch the healthy tissue. And we're seeing why this level of precision matters, especially in the brain. There was a surprising study this week linking senolytics to epilepsy.
Epilepsy, I would not have made that connection at all.
It's a very surprising link. The research indicates that senescent glial cells, these are the support cells in the brain, accumulate and drive the inflammation that triggers seizures in temporal lobe epilepsy.
So the inflammation from these zombie glial cells is the spark for the seizure.
It appears to be a major driver. When they cleared these cells in mouse models using senolytics, seizures dropped significantly.
So senolytics aren't just for anti-aging in the cosmetic or general sense. They could be specific treatments for epilepsy, for Alzheimer's, for osteoarthritis.
Exactly. They are disease-modifying therapies. But, and here is where the expert in me needs to urge some caution. We have to apply critical thinking. The kill-them-all approach might actually be dangerous.
Why? I thought we hated zombie cells. I'm ready to get rid of all of them.
We do, mostly. But biology is always more nuanced than we'd like. It turns out some senescent cells are actually necessary for wound healing and tissue repair. If you cut your skin, a temporary acute burst of senescence is part of the signaling pathway that calls in the immune system to fix the damage.
So if we wipe them all out, we might stop healing as well.
We might impair our ability to repair acute injuries. We need a Goldilocks zone, clearing the chronic accumulated senescent cells that drive aging, while leaving the acute ones that are just doing a specific job and will be cleared out naturally later.
It's never simple, is it? It's always about balance.
It always is.
Before we move on to the third pillar, the maintenance crew, I want to take a quick second for everyone listening. If you're listening on a podcast service like Apple Podcasts or Spotify, we'd be incredibly grateful if you could leave us a rating and review for Tomorrow Unveiled. It truly helps us reach new listeners and keeps these conversations going.
We do read them too. It's great to see what resonates with you.
Okay, let's get back to it. Segment 3, the code and the switch. We talked about resetting the software, but what about the hardware itself? And the physical switches in our body?
This is where we get into the next generation of gene editing. Everyone has heard of CRISPR by now, the molecular scissors.
Snip, snip.
Cut and paste. Right. But cutting DNA is inherently risky. When you slice the double helix, the cell has to repair it. And sometimes it makes mistakes. You can get off-target mutations or chromosomal rearrangements. It's a powerful tool, but it's a bit of a sledgehammer.
So what's new?
The news our team flagged is about CRISPR 2.0. It's a no-cut method.
How do you edit genes without cutting them?
They use what you could call molecular anchors. Instead of slicing the DNA strand, they use the CRISPR machinery, the guide system, to deliver a different payload and enzyme that can just remove the chemical tags that silence a gene.
So it's like unmuting a conversation rather than deleting the sentence?
That's a perfect analogy, and this is vital for aging. As we get older, many of our good genes that help with repair or tumor suppression or metabolism get epigenetically silenced. They get turned off. This tool could potentially turn them back on safely without risking genomic instability.
That feels much, much safer for a therapy you might take preventatively. Now let's talk about the exercise switch. This one. This one blew my mind. Piezo 1.
Piezo 1. This is the field of mechanobiology at its finest. I mean, we all know exercise is good for us. We know weight-bearing exercise builds bone. But how? How does the bone know you're lifting weights?
I honestly just assumed it was, I don't know, stress or magic.
It's piezo 1. To understand piezo 1, you have to imagine the cell membrane not as a static wall, but as a fluid dynamic sea. Piezo 1 is this massive propeller-shaped protein embedded in that membrane.
Propeller shape.
Yes, it has these 3 huge blades that splay out. When the membrane stretches, say, when your heel strikes the pavement while running, those blades mechanically lever open a central pore. It's not chemical, it's physical. It's a literal trapdoor that pops open under tension.
And what rushes through that trapdoor when it opens?
Calcium ions. They flood into the cell, and that spike of calcium is the starting gun. It travels to the nucleus and activates the transcription factors that basically say, we are under stress, reinforce the structure, build more bone.
So when we say weight-bearing exercise, the weight is literally pulling these tiny trapdoors open on our cells.
Correct. And this brings us right to the use it or lose it problem of aging and a sedentary lifestyle. If you are bedridden or just sitting in an office chair for 12 hours a day, the membrane never stretches, the trapdoor stays shut, the calcium never flows.
And the stem cell gets confused.
It gets the wrong default signal. Without that mechanical noise, that input, the mesenchymal stem cell differentiation pathway drifts. It switches from osteogenesis, which is making bone, to adipogenesis, which is making fat.
Wait, so my potential bone cells are actually turning into fat cells just because I'm sedentary? That is terrifying.
It's a huge reason why elderly people develop osteoporosis and marrow-adipose tissue at the same time. It's a mechanical failure. But the breakthrough our team highlighted is that they are looking at exercise mimetics, drugs that can chemically flip the piezo 1 switch.
So you could trick the cell into thinking it's running a marathon, even if you're bedridden after a hip surgery.
Ideally, yes. And this isn't just for people who don't want to go to the gym, though there's that. Think about the elderly who are too frail to exercise safely. If we can maintain their bone density chemically by flipping the switch, we could prevent the hip fractures that are so often the beginning of the end.
That is just incredible. Of mechanical switches to inflammation loops. We have to talk about GDF3. Our research team labeled this the inflammaging loop.
Inflammaging is a great term for the chronic, low-grade inflammation that just simmers in our bodies as we get older. It drives everything from heart disease to dementia. A major study this week identified a specific culprit, a protein called GDF3.
How does it work? What's the loop?
It's a particularly vicious cycle. Aging macrophages, your immune cells, start producing GDF3. But here is the critical design flaw. GDF3 then binds back to receptors on the very same cells that produced it. It's called autocrine signaling.
It's talking to itself.
And it's saying all the wrong things. It locks these immune cells into a permanent freakout mode. They stay inflamed and they lose their ability to fight actual infections effectively. It's a big part of why older people are so vulnerable to things like sepsis.
But the study showed a fix, a way to break the loop.
Yes. By blocking the pathway, specifically the GDF3-SMA23 pathway, they could break the loop. They reset the macrophages and reduce the systemic inflammation.
It really feels like we are finding the schematics to the machine. You know,
Yep.
We aren't just guessing anymore. We're finding the specific wires to cut or reconnect.
That is the theme of the week. Specificity. We're moving from blunt instruments to scalpels.
Okay, moving on to segment 4. We've looked at the cells, the genes, and the signals. Now let's look at the engine that powers it all. Metabolism. And this brings us to the superagers.
A fascinating group of people.
So we've talked about fixing the software with ER100. We've talked about clearing the waste with senolytics. We've even talked about chemically faking exercise. These are all interventions, things we do to the body.
External forcing functions, yeah.
Right, but then you have this study out of Brazil, the supercentenarians, people over 110. These are people who didn't have ER100. They didn't have senolytics. Most of them probably never stepped foot in a modern gym. And yet, they are thriving.
This is where the narrative completely flips. For years, the dogma was blue zones. We thought if you ate enough purple sweet potatoes and walked up hills every day, you'd live to be 100.
Or the other theory was that they were just genetic mutants who lacked the genes for heart disease or cancer.
They won the lottery by not having the bad numbers, right? But the genomic sequencing of this Brazilian cohort just destroys that theory. They have the bad numbers. They carry the risk alleles for Alzheimer's, for cardiovascular disease. often at the same rates as the general population.
So they're walking around with a loaded gun, but the trigger just never gets pulled.
Exactly. And that concept is what we're now calling biological resilience. They don't just avoid bad genes, they possess compensatory mechanisms. Maybe they have the gene that causes plaque buildup in the brain, but they have another gene that clears it out twice as fast. Or a gene that prevents that plaque from ever killing the neurons.
This changes the entire strategy for longevity medicine.
Completely. Instead of trying to gene edit out every single risk factor, which is practically impossible because there are thousands, we should be hunting for these resilience genes. We want to bottle that protection. We want to find out what makes them bulletproof and give that to everyone else.
Speaking of bottling protection, let's talk about some metabolic interventions that are a bit closer to home than rare genes.
Calcium alpha-ketoglutarate. It's a natural metabolite in the Krebs cycle. That's the central process for how our body makes energy.
The Krebs cycle. High school biology nightmares are flooding back.
I promise I won't make you draw the diagram. But CAG is really interesting because the new data shows it has a remarkable ability to reverse memory deficits, at least in animal models.
How does it do that?
It seems to bypass blocked receptors in the brain. In aging and in diseases like Alzheimer's, certain key receptors, the NMDA receptors, stop working properly. The brain tries to signal, learn this, but the receiver is off the hook. CAG seems to find a backdoor to restore synaptic plasticity, the ability to learn and remember, and it also activates autophagy, that cellular cleanup crew we talked about earlier.
And then there's mitopure, or urolithin A. This is getting a lot of buzz lately.
It is, and for good reason. Urolithin A is a postbiotic. Some people can make it from eating things like pomegranates, but most of us lack the specific gut bacteria to do so efficiently. The new findings are that it rejuvenates the immune system.
By fixing the mitochondria.
Yes. Specifically, T cells, your immune warriors, they need a ton of energy to fight infection. As we age, their mitochondria, their cellular power plants, get sluggish and damaged.
They just run out of steam.
Literally, urolithin A triggers mitophagy, which is the targeted recycling of old, broken power plants. The cell grinds up the bad mitochondria and builds new, efficient ones. The result is an immune system that acts younger, that's more robust.
It connects back to that idea of functional longevity. It's not just about looking young. It's about your immune system fighting off the flu like it did when you were 20.
Precisely. And briefly, we have to mention the ACCA drift in the gut.
The gut aging theory.
Yes, our research team highlighted a study that found that stem cells in the gut accumulate errors linked to iron metabolism.
Iron.
Yes. We usually think of iron as good, for blood. But excess iron or mismanaged iron causes tremendous oxidative stress. This study suggests that as our gut stem cells age, they get bad at handling iron, which leads to DNA damage. It links diet, iron levels, and stem cell health in a unified theory of gut aging. It's yet another lever we can potentially pull.
So we have the biology, we have the interventions, but how do we know if any of this is actually working in a specific person? This leads us to segment 5, the quantification of you.
The end of guesswork.
We saw a big announcement this week about using AI-driven imaging for longevity.
Yes, this is the democratization of high-end diagnostics. Previously, if you wanted a whole-body preventive MRI, you were probably a tech billionaire in Silicon Valley and you were paying $10,000 cash. Major imaging centers are now starting to roll this out as a specific service.
And it's not just looking for tumors, right? That's the old way.
No, that's the key distinction. Traditional MRI is reactive. It's looking for pathology. Is there a tumor? Yes or no? This is preventive quantification. The AI looks at the scan to measure things like liver fat before you get diabetes. It measures the volume of your hippocampus before you have dementia symptoms. It checks prostate density, muscle quality, visceral fat.
It's catching the smoke before there's a fire.
And it's giving you a number. You can't improve what you don't measure. If you know your liver fat is creeping up, you can intervene with diet or some of the metabolic agents we discussed, and then you scan again in 6 months to see if it actually worked. It turns your body into a manageable project.
And then there's this company, Nautilus. They're counting individual proteins.
Single molecule proteomics. This is mind-blowing resolution. Instead of taking a spoonful of soup, which is your blood, and sort of guessing the ingredients, they are counting every single noodle, every carrot, one by one. They're looking at tau proteoforms.
And tau is the protein that gets all tangled up in Alzheimer's disease.
Right. But tau isn't just good or bad. It has hundreds of different shapes and chemical modifications or proteoforms. Nautilus's platform can see exactly which specific forms of tau are appearing in the blood. This could allow us to spot different types of Alzheimer's years before symptoms arise, and perhaps match the specific type of disease to a specific drug.
True precision medicine.
Finally, we have to talk about the concept of organ clocks.
This was a big paper our team flagged.
Yes, we tend to say, I'm 45 years old, but biologically, you are a mosaic. Your liver might be 50, your heart might be 40, your brain might be 30.
Or vice versa, which would be much scarier.
The study found that brain aging is the single strongest predictor of mortality. If your brain is biologically older than your chronological age, your risk of death goes up significantly, regardless of your other organs. This suggests we need to move away from a single biological age number and start looking at an organ-by-organ dashboard.
And briefly, before we wrap up, what is Sidewinder? It sounds like a missile, but it's for DNA.
It's the printing press for the genomic age. It's a new method to synthesize to write DNA with incredibly low error rates, 1 in a million, which is a huge leap forward.
Well, why does that matter so much?
Because of scale. If we are going to build these gene therapies like ER100 or the aptamers for Sanalytics or the no-cut CRISPR tools, we need to be able to manufacture synthetic DNA quickly, cheaply, and perfectly. Right now, manufacturing is a huge bottleneck. Sidewinder is the tool that builds the tools. It's the infrastructure of this entire revolution.
So we've covered a staggering amount of ground. From rebooting eye cells to flushing out zombie cells, clipping exercise switches, and scanning our bodies with AI. What does this all mean when you put it together?
If you synthesize all this, what you're looking at is a combinatorial future.
It's not just one magic pill. It never is.
It never will be. It's a protocol. Imagine a future checkup, maybe 5 or 10 years from now. Step 1, screen. You get the AI, MRI, and the Nautilus proteomic scan. You find out your liver is aging too fast. Step 2, stabilize. You take a metabolic agent like Cake A or a piezo-1 agonist to shore up your body's natural defenses. Step 3, clean. You do a pulse dose of a targeted senolytic to clear out the inflammation and the zombie cells in that organ.
And step 4 is...
Step 4, reset. If the liver is really struggling, maybe you get a targeted injection of an ER100 style therapy to rejuvenate that specific tissue, to wind its clock back.
It's maintenance. It's just like owning a car. You change the oil, you replace the tires, you update the software.
And by doing that, we shift from talking about lifespan, just surviving, to the era of the biological citizen. We take ownership of our cellular data and manage it proactively.
Which brings us to our final provocative thought, a concept that came up in our research called bioliquidity.
This is a really provocative idea. If we accept that aging is damage accumulation and that young, undamaged cells are a finite resource, then your own biology becomes an asset, tangible asset.
Like a savings account.
Exactly. The concept suggests banking your young cells, your stem cells, your immune cells now, while they are as young and healthy as they will ever be. In a future where we can expand and reinfuse these cells, or use them for reprogramming, your own young biological material might become the most valuable currency you possess.
Bioliquidity. Investing in your future self. Literally. That is definitely something to chew on.
It certainly is.
We've reached the end of our time for today, but the conversation is really just starting. If you're finding this insightful, please take a moment to like this video, subscribe to Tomorrow Unveiled, and consider hitting that new hype button to help us reach more viewers. And of course, be sure to turn on notifications so you never miss an update on the future.
And we love hearing your thoughts. Whether you're on YouTube or your favorite podcast app, leave a comment and let us know what you think about the concept of bioliquidity and banking your own cells. Would you do it?
Until next time.
The future is closer than you think.